So, as you guys know, I am one of the co-founders of KTK, but also I am a grade 12 student attending Upper Canada College. Additionally, I have a passion for biology and medical science. So today I'll be talking about the research and development of CRISPR and its applications of cancer therapy. Furthermore, so my bio is that I went and represented Canada at HOSA IOC, which is an international medical competition. Lastly, I also achieved global silver in the British Biology Olympiads. Next slide. Okay, so CRISPR-Cas9 and applications for cancer therapy. So first of all, does anyone know what DNA is? Does anyone raise your hands? Yeah?
Like the gene, like the genetics in that.
Sure, yeah, exactly. So DNA is what encodes us all, the genetic information within us. So next slide. So basically what CRISPR is called a clustered regular interspaced short palindromal repeats. So basically what I call this is a library. Basically this is where cells retain all their information of bacterial DNA. So whenever there's a virus that affects or implements or hijacks the cell through bacterial DNA, the cell will be able to use CRISPR to take out the bacterial DNA and use it to record that virus, learning how to protect and defend itself over time. So here we can see, using this, we can use CRISPR. I'll show you the next slide. So, how did CRISPR start? CRISPR started from a basic research project, learning how bacteria are able to defend against viral infections. Through this, they were able to learn that CRISPR had an adaptive immune system, which was called CRISPR. And using this, they were able to detect and destroy the viral DNA and collect it overall. So what did the scientists see in CRISPR? Usually, it's because of this thing called the PAM. What the PAM did was able to seek out a specific sequence of gene, which was the bacterial DNA that was encoded inside the DNA sequence. So through this, they were able to seek out and cut the viral DNA, which is what made it really interesting for the researchers. Next slide. So here you can see the Cas9 and the DNA and RNA. Does anyone know what an RNA is? RNA? No? Okay, so an RNA is an intermediate type of sequence of DNA. Because you know DNA has two sequences, right? An RNA only has one sequence, meaning that it only has one strand, compared to DNA having two strands. What this does is allow the Cas9 to create a copy of the RNA and transcribe it. Next slide. Jerry. So here, by cutting and replicating the RNA, they're able to use that to cut it. Let me show you a deeper insight into what it does. Here, through this, they're able to find the DNA, which is basically the bacterial DNA, and cut it using the scissors they have, which is called the PAM. And by doing this, they're able to replicate it and repair it with new genetic information. Okay. Furthermore, the CRISPR-Cas9 also has a guide RNA. What this does is also target its RNA sequence. Basically, after the CRISPR replicates the DNA, it's able to seek out the RNA sequence. And by doing this, the PAM is able to detect the specific sequences in the DNA. By doing this with the cutting domains, it can change the entire amino acid. Does anyone know what an amino acid is? No? Yeah, so an amino encodes a protein. By using 3 types of specific amino acids, we're able to create a protein. Next slide. So here, by doing this, we're able to see, let's pretend AGCTAG is a DNA sequence. And by replacing that with CRISPR, we're able to replace that sequence with something else, because we're able to locate and fix onto that sequence and hijack it By doing that, we're able to replace that with something maybe like ctCTAG. Does anyone know why we want to do something like this? No? Basically, what we can do with this is to change gene expressions, like what we said are proteins. Basically, gene expressions are like, for example, our hair color. Our eye color, or maybe even our skin color. Little special proteins like this can encode and change everything about ourselves. Next slide. So, let me summarize this about what CRISPR does. Firstly, it binds using the PAM to find a specific sequence. Using the specific sequence and replicates it into an RNA. Secondly, it then uses the cas9 complex to double and break that sequence. Thirdly, it uses cas9 to remove and repair the sequence overall. So then basically what CRISPR does is replicating Cas9 breaks it. So this creates a lot of opportunities for us, such as sickle cell anemia. We're able to change these sequences because of people are born with these like mutations that change their, let's say, okay. So basically, let's say some mutations, we're able to change that now with CRISPR by scientists being able to see these DNA sequences and see there's a mutation. For example, it should be CTAG, not CTGG. They can change that G into an A, which is an amino acid. So this creates many opportunities. Next slide. So what are some current applications of CRISPR? So who here likes tomatoes? Yeah? Tomatoes?
Yeah.
What do you like them on?
Pizza.
Pizza? Pizza?
Burger.
Pasta. Pasta? Nice. So, have you guys ever thought about making tomatoes spicy?
No.
Cherry neck. Spicy, yeah. Spicy pizza? Yeah. So in some worlds with conditions that cannot make spice, like curry spice or something like that, right? We can now change a gene sequence in tomatoes to make them spicy, making them being able to be palatable and delectable. Next is mosquitoes. Who here hates mosquitoes? Boo. Yeah, exactly. Everyone here hates mosquitoes. Because first off, did you know they kill over 700,000 people per year? Yeah, exactly, 700,000 due to malaria and its infections. So, some researchers from the Imperial College of London were able to find that by changing a specific sequence in mosquitoes, they were able to prevent them from laying eggs, overall slowly diminishing their population. Next. Who here ever thought about going back in the past to see some maybe, George Washington or something like that? Yeah, or seeing their favorite idol. Or maybe Michael Jackson, who knows? But here, instead of bringing to them, or maybe it's like or seeing an animal, like a mammoth. Who here wants to see a mammoth? Yeah. But what if instead of bringing us to them, we bring them to us? So basically, by finding these specific characteristics, like tusks, and their skin, we're able to use those characteristics and implement them, change the sequence to their, like say, descendants, like the elephants, by having elephants with woolly skin and such, creating this type of hybrid animal. Okay, next. So, how does CRISPR-Cas9 eliminate cancer therapy overall? Who here has a guess on what we can do to use CRISPR to eliminate cancer?
Radiation.
Radiation? How's that?
They change the cell.
Oh, yeah, exactly. So people usually think about using, good job. So, people usually think about, since cancer is so complex because of these mutations and its DNA sequences are changed, right? By using CRISPR-Cas9, we're able to change those mutations back into regular DNA sequences. Well, that's what most researchers thought. Next slide. Unfortunately, this is not achievable because cancer is a mutagen, and basically what happens when researchers find out by changing the sequences, we're able, the cancer and the tumors are actually changing mutations again and again and again. So basically, by doing that, by using CRISPR to change the DNA sequence, it actually exacerbated the issue, making more mutations and increasing the tumor size overall. So, and additionally, as I said, it's really expensive. CRISPR is really expensive to use and is also very pricey. It's also not cheap and easy to use, as I said. Next slide. So, what did the researchers do to use CRISPR? Basically, what they did was use something called a T cell. A T cell is a type of white blood cell that attacks viruses and such. And using this T cell, they're able to kill cancerous cells, but they're not too efficient. So hence, the researchers used something called a CAR T cell. Basically, they attached a CAR, which is called a chimeric antigen receptor, onto T cells to make it more efficient. So make it more faster, making it a super white blood cell that's able to kill cancers way much faster and efficiently. So basically, how a cancer T cell kills a bacteria is by binding to the specific cell and killing it overall, because usually cancers are cells that cannot die. So here we see is a T cell, and by using, like I said, CRISPR to change the genes and attach a CAR receptor, we're actually able to make it a super white blood cell that then kills overall the cancer cell. And here we can see how they do that. So they isolate the tumor samples, so taking the tumor out of maybe a mammal or such, and attaching that to a T cell, and injecting that solution back into the human body to overall kill the cancer patient, the cancer inside the patient. So that's it. Thank you all for listening.